From the moment you swallow, food enters a single, continuous tunnel, stretching from the oral cavity down into the abdomen. This upper segment of the gastrointestinal tract includes the oral cavity, the pharynx, the esophagus, and the stomach, terminating at the border of the duodenum. This single pathway is required to perform contradictory tasks. In a matter of inches, it must withstand physical friction, secrete acid to break down proteins, and then neutralize that same acid to prevent tissue damage. To understand how one tube executes these different tasks, we have to look past the macroscopic organs and examine the microscopic architecture of the tissue wall. Despite the different functions performed by each segment, the entire gastrointestinal tract is constructed from a universal 4-layer histological blueprint. By mastering this blueprint and tracking how its layers adapt to local needs, one can identify any segment of the gut and deduce its physiological role. This baseline model of the gut wall begins with the innermost functional layer, the mucosa. The mucosa consists of 3 specific subcomponents: the lining epithelium, the underlying connective lamina propria, and a thin boundary of smooth muscle called the muscularis mucosae. The second major layer is the submucosa. It is composed of dense, irregular connective tissue, which provides the structural integrity needed to support the tube. Surrounding this is the third layer, the muscularis externa. By default, it consists of 2 muscle orientations, an inner circular layer and an outer longitudinal layer. The structure is completed by the outermost boundary, the adventitia, or the serosa if the organ is invested by the peritoneum. This architecture is controlled by the enteric nervous system. Within the submucosa, the submucosal, or Meissner's plexus, coordinates localized glandular secretions. A second network, the myenteric, or Auerbach's plexus, is located between the 2 muscle layers and drives the mechanical contractions of peristalsis. This 4-layer architecture, with its embedded autonomous controls, is the template upon which every localized adaptation of the gut is built. In the esophagus, the primary function is the rapid, protected transit of food from the pharynx down to the stomach. To withstand the intense physical friction of a swallowed bolus, the esophageal mucosa is lined with stratified squamous non-keratinized epithelium, multiple layers of flat cells stacked to act as a barrier. The esophagus also contains specialized glands within the submucosa. This is a distinguishing feature, as the submucosa remains devoid of glands in nearly all other regions of the tract. The muscularis externa here follows a specific gradient. The upper third is composed of skeletal muscle, though it remains under involuntary control during swallowing. The middle third contains a mix of fiber types, while the lower third transitions entirely to smooth muscle. This musculature forms the upper and lower esophageal sphincters, which maintain one-way movement and prevent gastric acid from entering the tube. These mutations, the friction-resistant lining and submucosal lubrication, adapt the base blueprint into a transit conduit. As the pathway enters the stomach, the structural requirements immediately change from transit to storage and chemical breakdown. At the gastroesophageal junction, the epithelium shifts. The thick layers of the esophagus are replaced by a single layer of tall, simple columnar cells. The mucosa invaginates to form deep gastric pits. These channels lead to expansive gastric glands in the lamina propria, designed for high-volume secretion. Within these glands, chief cells secrete pepsinogen, the precursor enzyme used to begin the digestion of proteins. Parietal cells secrete both intrinsic factor for vitamin B12 absorption and the hydrochloric acid required to activate pepsin and destroy incoming bacteria. Scattered throughout the mucosa are enterochromaffin-like cells, which release histamine to promote this acid secretion. In addition to these chemical secretions, the stomach requires a specialized mechanical structure to physically churn food into chyme. To achieve this, the stomach adds a third, innermost oblique muscle layer to the muscularis externa, providing a new axis of contraction. By deepening the mucosa for chemical output and adding a third muscle layer, the stomach becomes a chamber specialized for maceration and digestion. Moving past the pyloric sphincter, the duodenum marks the transition into the lower digestive tract. The mucosal surface morphs from the flat pits of the stomach into finger-like projections called villi, which increase the surface area for nutrient absorption. This region faces a specific threat. It must receive the highly acidic chyme directly from the stomach. The duodenum's defense is a high concentration of Brunner's glands located within the submucosa. These glands produce alkaline, mucoid secretions that neutralize the gastric acid as it enters the small intestine. These specialized glands act as a checkpoint, neutralizing the chyme to protect the absorptive tissues that follow. Because these structural adaptations are consistent, observing a microscopic tissue slice allows for the identification of the gross anatomical organ. In the esophagus, we look for stratified squamous epithelium protecting the lumen alongside glands located in the submucosa. In the stomach, we find simple columnar epithelium with deep pits and 3 distinct layers of muscle in the muscularis externa. In the duodenum, the diagnostic markers are the projecting mucosal villi and the expansive Brunner's glands in the submucosa. Throughout these segments, the enteric nervous system remains a constant, coordinating the specific secretions and contractions required by each region. The upper gastrointestinal tract demonstrates how a single architectural blueprint is modified to transport, digest, and neutralize food, preparing the way for the complex absorption of the lower bowel.